Welcome everybody to the Genomics Powering Sustainable Mining Water and Mining Water Futures, proudly presented by the Environmental and Social Responsibility Society, ESRS. During the webinar, if you have any questions, please feel free to write your questions in the question box. Matthew Hutter and me, Marcia Forche, we'll try to address your questions after, and if we can't answer during the webinar, we will try to follow up with answers for you. Just a little bit of a blurb on us. So ESRS, our vision is to provide leadership and professional development opportunities as they relate to social and environmental responsibility and performance of the Canadian mining industry. A couple of our upcoming events is the ESRS quarterly meeting, October 5th, and we'll have monthly meetings. I'm Marcia Forche, and the other moderator is Matthew. Just a brief land acknowledgement. I'm presenting or I'm moderating from Saskatoon, Saskatchewan. So we have Treaty 6 and the homeland of the Métis. And just a quick look at a safety moment. Everybody has been working from home on and off, so just be aware that you can have sustained awkward postures that can lead to lifetime injury or pain. Workers should regularly stretch, exercise, do strength training to avoid ergonomically induced injuries. And I'll hand it over to Matthew.
Perfect. Thanks very much, Marcia, and good day to everyone. So today we'll be exploring some new developments in microbial characterization and how they relate to the mining industry. Although mining systems are critical for industry across the globe, applying these new characterizations within the mining industry has been relatively slow. So today we're very fortunate to have our guest presenter, Professor Lesley Warren of the University of Toronto, who will take a deep dive with us today into this topic. The presentation itself is scheduled for approximately 35 to 40 minutes, followed by a brief 15-minute or so question period. The webinar can continue in past this time if there's interest to continue asking questions, but I will chime in once we hit the official hour mark. As Marcia mentioned, please submit your questions in the panel on the right, and we'll keep track of them for the Q&A. And now to officially introduce our presenter. Professor Lesley Warren is an internationally recognized, innovative researcher delivering new water management and treatment technologies through novel discoveries of the mining microbiome. She moved to the University of Toronto to take up the Claudette Maquet-Lasson Chair in Mineral Engineering in 2016 in the Department of Civil and Mineral Engineering. She also became the first female director for the Lassonde Institute of Mining in 2018. She's established a pioneering multidisciplinary research program investigating the roles of bacteria in key processes affecting mining and resource sector waste environments. Over the last 10 years, Dr. Warren has brought in millions for energy sector and mining industrial partnered research, trained over 50 highly qualified personnel, and given over 40 keynote talks, including presentations to MPs and senators on Parliament Hill. I completely filled the slide, but I could probably go on even more. But I guess without further ado, I'd like to present a warm welcome to Professor Warren and introduce her to share her presentation now. Welcome.
Thank you very much. I'll just pull up my presentation. Here we go. And it's an absolute pleasure to be here, and I want to thank everyone for attending. I know genomics is not a word that always falls easily off everyone's tongue, but it's just a simple way to talk about bacteria. And really the revolutionary technological transformation that's happened in molecular biology has been slow to be adopted in the mining industry. And so what I want to talk about today are a couple of things around essentially demonstrating the validity of using these techniques and why they're so relevant. So we know that, and we're seeing this already, that global water demands are outstripping available supplies. Mining around the world is facing increased scrutiny because they are a significant, often the largest user for regional watersheds. and their wastewaters can pose massive risks to environments and communities if they are not managed appropriately. And we know that, sort of with the energy transition, that mining intensity is only going to increase, and that puts even greater pressure on water. And the irony is that we may be looking at climate solutions from an electrification perspective, but if we thoroughly degrade our freshwater resources in the process, that is not going to be very successful. So, there are increasing operational and reputational risks. And really, this is where we're looking for what are some of the innovations that are being applied in other sectors that would be relevant to start thinking about how can we be better at managing using water supplies in the mining sector so that they use less, they steward it better so that there are fewer, if hopefully no, impacts to surrounding environments and communities. And so microbiome is a word you may or may not have heard of. It simply refers to the complex microbial communities that occur in an environment. These microbiomes are contextually distinct. So what that means is if we take a sample from the soil or from a doorknob or even from your hand, those organisms will be distinctly linked to the specific characteristics of that environment. And they play key roles in shaping these contexts. So they're not simply there, they're actually interacting and engaging with those environments. And over the last 20 years, since the molecular biology revolution began, we've really seen transformative new insights into processes at the macro scale. So soil nutrition, ocean carbon sequestration, cancer, and most recently, obviously, pandemics. And I think that, there are two things that really go into this. One has been the demonstration that the application of these technologies and understanding which microbes are present, what are they doing, what are the outcomes, really better informs understanding. But secondly, our resolution, those technologies have gotten so much more powerful and so much cheaper. So what took billions of dollars to do 20 years ago, when they did the first human genome mapped completely, would now take a few $100s. And you can have those results in a couple of days. So it's far more accessible, and I think it's far more powerful. Having said that, and we've seen the sort of microbiome, this exploration of microbial communities, microbes in any environment outside of the Petri dish do not live by themselves. They live in communities with other microbes who are often doing different things. So they work essentially like a large macroorganism. And so that work has been going on all over the world, all over the planet. And typically researchers will put those data, identifying the organisms and what they do, into public databases like the NCBI. We did a recent search of that NCBI, which is one of the largest public databases for genomics information. Mining systems investigation is less than 1% of all of those studies that have been published. And so what this really tells us is that mining wastewaters and mining waste contexts are really microbial black boxes. And the relevance of this for anyone who's working in the environmental sort of section for a mine knows that there are commonly times when predicted model predictions fail. You have non-compliance off-site. Your tailings pond starts to do things that you were not expecting it to do. And that's always a sign that something is missing from your model. And we've seen this, in so many other systems now, whether we look at soils, whether we look at oceans, lakes. We know that to understand what's going on in these contexts, you have to think of the interactions, not only of the rocks and the water, essentially think about the minerals and the elements that are present, but you now start to have to add the microbial catalysis of certain reactions. And bioreactive elements, such as oxygen, sulfur, nitrogen, iron, manganese, metals, these are all very prevalent in mining wastewater systems and often at high concentrations. And so we can start probing questions about who's there, what are they doing, how do they do it, what are the controls and outcomes? And that simply provides a much more solid foundation of understanding that's proactive. What are the processes that could happen? What are the controls on those processes? What are the outcomes? And so this systems-based investigation is really around filling in those knowledge gaps that are contributing to the risks that most mining companies are facing right now in environmental management. And in particular, mines that are processing sulfur sulfide-containing ores, which is quite common. 70% of the metals globally come from sulfide minerals. Commonly can have, if they're not well managed, impacts in the receiving environment, such as acidity, oxygen depletion, toxicity, contamination. And that's simply because, well, there's a couple of things. One is sulfur chemistry is a little bit challenging, it's complex. Some sulfur compounds are recalcitrant, will pass through treatment. And secondly, the bacterial processes that may be catalyzing some of the cycling of those sulfur compounds, generating new compounds, which are generating more acidity or are more recalcitrant to treatment, are not constrained at all. And so one of the reasons that we got involved in this project was a couple of mining companies approached us because this thiosalts question, this word that is common in the mining industry for these sulfur compounds, common in tailing streams from mines that are milling and processing sulfide ores, was that they were starting to see these challenges. And so this is just a picture at the bottom of a tailing stream being deposited in a tailings pond. And when we do some epifluorescence microscopy, so we take some of those tailings, we probe them with fluorescent probes, which light up if essentially life is present. You can see that that pyrrhotite particle, that sulfide particle, is entirely coated by life. And so life becomes the interface between the particle and the water. So what those bacteria are doing will absolutely influence what's happening in the water. And so, you know, the risk from a business perspective is that you have reactive sulfur compounds in tailings ponds. They're complex, they're challenging, and we don't know everything about them. A lot of them are recalcitrant to treatment if they get into the receiver, which they can do, and not all models are predicting when that's happening. You can then have oxygen consumption, which leads to fish kills, acidity, again, which damages the receiving environment. And this, of course, is an industry liability and risk. And again, it simply highlights that there are analytical, most likely, and microbial knowledge gaps that are really decreasing the efficiencies and limiting the tools available to managers who are trying to manage their waters and wastewaters most effectively. And so the Mining Wastewater Solutions Project, you'll hear me refer to it as the MWS project, was really an industry academic partnership. It was really driven by Hudbay and Glencore Sudbury INO, and we brought Rambler on board as well. A multitude of people because we're doing multidisciplinary research. We had a number of consulting companies, EcoReg and Ecometrics in particular, who really supported the work. And really it was beneficial because as an academic, we have ideas about how a system could work and how to do the analyses and the investigation, but being informed by mining partners who can really speak to the history, the legacy, what are the processes they use, really ensures that the research is targeted and it hits the right questions so that the answers we derive are actually valuable. And we started with very simple questions. And there was one that was simply, you know, do the current thiosalts methods, which are typically for most mining companies, they're sending samples to analytical labs off-site. Do those methods capture appropriately and adequately sulfur risks that are present in a tailings pond? And if not, can we develop new approaches to better monitor sulfur-based risks? Secondly, when we start to get into the microbiology, can biological indicators, whatever sort of level of genomics we want to use, and the simplest would always be the best, can they be more sensitive? Can they pick up essentially alterations in water chemistry or predict changes in water chemistry that would be relevant for the operator to know? And finally, can we start thinking about biological monitoring? So what would be the tools that would allow us to monitor key organisms that we know would signal specific changes or conditions? And can we start thinking about ways to use those endemic organisms, ones that are already present in those tailings ponds and are quite happily living there? Can we think about ways to manipulate them as treatment solutions? So the 4 mines are spread across Canada. What's interesting is that they're all base metal mines, but they vary in climate, they vary in age, size, and target metals. And the way we approach this, we work with a number of different companies in different projects, but we're always looking to understand those water systems essentially as a system. So that means we're going to look at the physicochemistry. What does the pH, the oxygen temperature look like from the top to the bottom? What's relevant geochemical parameters? But we're also going to include in that the microbiology. And particularly if we look just quickly, the total sulfur, if we go from Flin Flon, Hudbay's sort of oldest mine, 1927, down to Rambler, 2009, you can see that the older mines have more sulfur in their systems. So we think in millimoles just because it's easier to think about reactions and balance them. But 10 millimoles of sulfur is about 320 milligrams per liter of sulfur, which may not seem high in a mining context, but is very high in an environmental context. And you can see that they range a little bit in DO, but there's always some DO present, but never 100%. And particularly most of the time, most of these tailings ponds are circumneutral pH, which is quite common. As I mentioned, we do sort of an investigative, integrated approach. So we're looking not only at spatial sort of variability, what does the top of the tailings pond have going on, what does it happen at the bottom, but also over time and over space. And we're collecting all of these samples and processing them. So we really have a rich systems-based understanding of what processes are happening when and where, what are the controls and what are the outcomes. And so if we get to this first question, which took us a while to really understand what the analytical challenges were, working with analytical labs and then mining companies and academics, there was a little bit of language that we had to sort of work through. But we knew from our partners that, and indeed from the Mining Association of Canada, who's had several sort of thiosalts working groups, that total thiosalts measurements, the way they were being done, were insensitive to potential outcomes, and that's never a good thing. So if you can't predict what's going to happen to your receiver and then you have a non-compliance, that is not a good outcome. And there were time delays. So getting the results back meant that you were not able to adapt your management, because if it took 3 or 4 weeks from the time you took the sample, your window to actually intervene is gone. And secondly, the treatment requires chemical, energy, and infrastructure. It's a static approach. That's not necessarily the most sustainable approach, and it wasn't 100% effective. And so it was very clear that there was a need to start understanding how to improve that system. So when we started to dig into this, it was very clear that, there was a practical logic to this, was that essentially all of the thiosalts methods that were being used were really looking at only 2 potential sulfur species, thiosulfate and tetrathionate. And that's because they're very common in tailings lines from sulfide ore milling, and they're often the dominant sulfur species. And so those assumptions were, this is what's coming out of the tailings, so that's what's going to dominate in our tailings ponds. If we estimate those, we're good. And so when we start to, and of course, if those sulfur species are oxidized, that's what leads to generation of sulfate, but more importantly, acidity. But if we actually look at sulfur, there are a whole bunch of other potential sulfur compounds, which we would call under the collective umbrella reactive sulfur species, because meaning that any of these, if they're present, their oxidation can lead to acidity. So if you're not measuring them and they're present, they actually could be a risk to downstream receivers. And so we wanted to investigate, do in fact current thiosalts measurements, which are really focused only on thiosulfate and tetrathionate, are they essentially measuring all of the reactive sulfur or compounds present in sufficient quantities that would really suggest this method is not conservative of risk? So the way we looked at this was to do a mass balance approach. And so the bigger sort of circle is all the sulfur that could be in a system. Some part of that is likely to be sulfate. And then what we consider to be the teal, the second circle, the reactive sulfur, would include things like thiosulfate and thiosalts. And so the question is, if that sort of orange and lighter green circle, if they essentially comprise all of the reactive sulfur, then there's something else going on in terms of why models are failing to predict outcomes of their mining wastewaters. But if they don't, then it says what actually is happening is that you are not actually capturing all the sulfur that's in your wastewater that could pose a problem. So using this mass balance approach, and I'm just showing some select data here from one sampling event on one mine tailings pond and its receiver. And so you can see on the left-hand side, as you're looking at your on the screen, on mine in the tailings pond at three different depths, you can see that in all cases, the reactive sulfur, the teal bar is significantly higher than either of the other two methods. So what we did was we collected samples, we sent them to the analytical labs that our mining partners were using to get them measured for bulk thiosalts and thiosalt, and then we did the reactive sulfur method. And so you can also see that essentially there is sulfur that is making it through treatment into the receiver. So this also speaks to not only are these methods not capturing all the sulfur that's present, but the treatment is not actually able to get rid of it. And when we look across mines, and again, sort of looking again now, if we look at speciation and we look at sulfur reactive, this S-react value, which is just the total sulfur minus sulfate. So we take all the sulfur atoms, we measure them, sulfate is the end product, subtract that, any sulfur atom in any compound that's left is potentially reactive in terms of being oxidized, generating acidity. And when we look at these sulfur react speciation, so what sulfur compounds are in there, we see that in fact, ones that would be considered thiosalts, so things like tetrathionate, sulfite, or thiosulfate, really are not a significant component of that. And so this is, we have 48 samples. There's a paper that came out of this across 4 mines. And in particular, thiosulfate, which is commonly sort of focused on, only accounts for about 4% of the overall sulfur budget. So it's often quite low. And so it really sort of showed that the current methods, the inefficiencies, and also that mining companies were not aware that depending on which lab they were using, they were getting numbers that were only thiosulfate or tetrathionate. And so they were sometimes comparing apples and oranges. So there was a lot of clarity that came out of this particular piece of research. And it also showed that, this is also why they were not always predicting what was happening in their receiver, because we could see that there was sulfur making it through in the receiver, but it would not have been picked up by those methods most of the time because it would fall into essentially the sulfur pool that they were not measuring. You'll also notice that we've got a piece here on the previous slide and on this slide in terms of the unresolved SOI pool. And what we mean by that, so SOI is sulfur oxidation intermediate, so any compound with a sulfur oxidation state less than sulfur and sulfate that's capable of oxidation. What this means is that we still don't know, even with the speciation that we do, which is a little bit more complicated, we still don't know what all of this sulfur is, and there's ongoing work trying to figure this out, because obviously the more we understand what sulfur compounds are present, the more we can target and refine our strategies for management and treatment. And so the bottom line was that, this really sort of spoke to the fact that there were undetected sulfur risks passing through not only, one mine's treatment system, all of the 4 mines in the study, we were seeing this happen. So this really spoke to the fact that we needed better methods. As I mentioned, this came out, I think in 2020, and this mass balance tool, the advantages of this, it's very straightforward. A lot of times you have the data if you're working on a mine in terms of sulfate and usually total sulfur, and if you're not getting it, you can ask for it in the suite of metals when you're getting your analyses done, and then it's just a subtraction. So it's a straightforward number to get. And so what we're really delighted about was that Hudbay, who has been a tremendous partner on this project, managed to get regulator approval very quickly to use this mass balance approach for reporting sulfur. And so since January of this year, they are now using that approach. We're working on another method to essentially do rapid determination of sulfur react on site that would be easy for mines to adopt. And we're sort of hoping to beta test that later this year. I think there's great promise to that. And then working through with CSIRO in particular, who's an analytical chemistry lab based in Sydney, Australia, a very strong group in terms of sulfur chemistry. So we're looking to see if we can delve into the sulfur speciation a little bit more and identify what some of these unresolved sulfur species are. But I think in terms of considerations of this, it's conservative of risk, fast turnaround, simple, very simple methodology, and it's economically feasible. To do the mass balance approach is much cheaper than what you're paying analytical labs for currently. So that was a huge success, and it also gave us windows into understanding more about the sulfur chemistry, and that really did speak to we needed to we were confident from the beginning that microbes were going to be important, but the fact that there are other sulfur compounds present, and often present in higher concentrations than those ones coming out of the tailing stream, spoke to a significant sulfur cycle happening in these tailings ponds. And so the question is, if we start to delve into that, could we come up with better indicators, more sensible and sensitive indicators, and sort of possible risks in a way that would give mines time to adapt and intervene to prevent problems? So looking at a genomic baseline is basically just using molecular biological tools. And we can do this for any kind of sample: water, soils, rocks, tailings, doesn't matter. Grab a sample, we extract the microbes, usually by filtration, and then that filter has essentially captured all the microbes. We can extract then the DNA, the RNA, and then you basically run that through a sequencer and do all kinds of fancy computer stuff. What's the bioinformatics is essentially identifying organisms, genes, and then putting that story together. And so we can ask questions with 16S. You may have heard this term before. 16S for bacteria is essentially genes of heredity. So we can use that to ID different organisms, so different genera that might be present. Metagenomes, we're looking at all the genes in the community. So which pathways are possible? Not just genes of heredity, genes of function. So how are they making a living? Then we can also look at, with RNA, metatranscriptomics, which genes are actually active in the community. And that really starts to pinpoint, they might have a metabolic repertoire that allows them to do X, Y, and Z. But by doing the transcriptomics, looking at which genes are active, we see actually they're only doing metabolism X at that time. So it really allows you to discriminate from who's there, stamp collecting, to what could they possibly do. Interesting, I have a potential model, to actually what are they doing under given conditions. And so it's very powerful to bring into an integrated biogeochemical investigation. This is a complicated graph figure, and really it's to point out that not only is sulfur speciation complex, we have multiple potential sulfur species, but the genes that are involved in catalyzing reactions. So a microbe that's oxidizing a given sulfur compound, like thiosulfate, depending on which genes it has, will catalyze a different reaction with a different sulfur outcome. But these are organized in our best understanding to date. And as much as sulfur speciation still needs work, it's not just in mining context, it's in all environments. We don't have a handle yet on all the sulfur compounds that can occur. Understanding how organisms oxidize sulfur, we're realizing, again, this is a developing framework. But what we often see is the complete SOX pathway. You'll hear me talk about that. One of the interesting things about the complete SOX pathway is that typically organisms take it all the way to sulfate, and you tend to see acidity generation. The other one that's of great interest is the RDSR pathway. And in this one, this is where we get recycling of sulfur in this intermediate pool. We get disproportionation. The bottom line is organisms that are still gaining energy by using sulfur, following the RDSR pathway, don't produce as much sulfate and acidity. There's a lag to that. They tend to recycle compounds, and you see the generation and alteration, which speaks to why we might be seeing differences in the sulfur compounds occurring in a tailings pond compared to what came out of the mill. And so when we sort of put that together, one of the first things we really wanted to understand was that, are there distinct sulfur oxidizing bacteria that occur across mine, tailings ponds, and receiving environments? So are they the same? Is there sort of a bunch of guys that we find no matter whether we go to Hudbay or Sudbury INO? And that's important because then that starts to speak to a distinct set of organisms that are linked to mining wastewaters that we can really start to understand. If we understand what they're doing in these contexts, probably those results are more broadly relevant across lots of mines that are processing sulfide ores. And so we are really happy to see that, in fact, that is the case. What you're looking at here with the two pie diagrams are essentially anything that's yellow, orange, brown color. Those are sulfur oxidizing bacteria organized by families. And we had 7 major sulfur cycling families that occurred across tailings reservoirs. So the average, you can see sort of on the left for tailings reservoir, but also pretty much the same organisms appearing in the receiving environments, though in different proportions. And so what this really sort of told us was that we had sort of, if we understood what these guys were doing and how they did it, under what controls for these 4 mines, it was probably going to be relevant to other mining contexts. What's also interesting is that many of these sulfur oxidizing bacteria, these SOB, are novel. So they're not in public databases. And what that tells us too is that there's a really distinct set of organisms that are adapted and evolved to be in these kinds of contexts. And as we start to see patterns of these guys in these mining contexts, they are not going to be the same as what we might find in an ocean or a hot spring, where there's also often, sulfurous organisms. So the discriminatory ability of these kinds of results really speak to a distinct set of organisms really associated with these mining contexts. And again, this just sort of looks, if we look at the top, by abundance, 10 genera that occur across all of our mines. The dotted line just speaks to the maximum abundance, and you can see in some cases they're really high. I mean, in the tailings pond at Sudbury INO, our maximum abundance is almost 100%. So very, very highly adapted, efficient sulfur oxidizers going on in that system. But you can see that there are sulfur oxidizers in all our systems. And in fact, and I'll speak to this green versus yellow business in a minute, but you can see that the green guys are present across all our systems. And then we sometimes have these yellow and orange, and that becomes significant as I sort of delve into the story. So we know we have a distinct SOB mining water microbiome, right? This community that's really distinctly associated with mining wastewaters, and incidentally very different than what you would see in ARD contexts. But now what we want to know is, does that matter? Does it matter if they're a green one or a yellow one? Like, what does that tell you? And so for that, what we really needed to do was we started collecting enrichment. So every water sample we've got, and so far we have over 350 since I created this slide. But at the time, we have every sample we would create a sulfur oxidizing bacterial enrichment. We would select for those sulfur oxidizers, and then we could start experimenting with them. So we could take them from our 4 mines, we could grow them under the same conditions and look at how they process sulfur and what were the outcomes. And that's exactly what we did. And this particular study, I think, is a really interesting one, because what we did, so what you're looking at for each of these different bars that represents a different water sample, whether it was collected from a tailings pond or a receiving environment from the 4 mines. But what you're looking at is how much acid to sulfate they generated when they were grown under the same conditions. So what this tells you is that even when I grow them under the same conditions, there has to be something different in those original parent communities, because they're not all doing the same thing. And so what this came down to was essentially, if you tended to have more of the yellow and orange organisms, we saw more of the sulfate acidity generation. If you had more of the green guys, they were still processing sulfur, but they were generating more of these intermediate compounds, not generating as much acid and certainly not as much sulfate. And so clearly, how much acid and sulfate you get is driven by which sulfur oxidizing bacteria are present. And that really then started to get us thinking about this idea of slow SOB and fast SOB. Because again, if we go to thinking about managing a tailings pond, if you've got fast SOB on site, it's actually an advantage, because they theoretically could convert all of your reactive sulfur to sulfate. You deal with the sulfate and the acidity before you discharge, but you're not actually discharging any reactive sulfur that could cause issues in your receiver. However, if you've got more of the slow and they're generating more of that reactive sulfur pool that's not thiosulfate or tetrathionate that you're measuring, you're actually not even aware that they're there, and this is what they're doing. The potential for that to move off site and then cause a problem is higher. And so we started looking at them as fast and slow, and I'll sort of say, well, why, what's driving that? And it comes back to, for those of you guys who are thinking ahead of this, it comes down to what genetic machinery do they have. But I think what's also important, and we often get asked this when we're running lab experiments versus what's happening at site. And so what was really interesting when we went back and looked at our field data, this is one of our mine sites, just looking at a time series. And you can see here that we're looking at the relative abundance of slow, so the green guys, versus fast. And Halothiobacillus tends to be our star sprinter when it comes to fast in our mining context. But you can see that when Halothiobacillus dominates, we had acidic pHs in those tailings ponds for the 2 years they dominated. However, when the slow guys came back, then we see the circumneutral pH. So this experimental result played out in the field data when we went back and looked at it, and it also played out in the receiver. So again, we saw that where we had more of the fast guys, these yellow-orange guys, then the pHs in the receiver tended to be lower. And so this, was great because we were able to show synergy between what we were seeing in the lab and what we were seeing in the field. And what it comes down to, and Lauren Twyble, who's one of my PhD students, who's been involved with the project pretty much since word go, she'll have a couple of really good papers coming out this year, but really started to look at the metagenome. So what genes do all of these sulfur oxidizers have, and how can we start organizing that in a way that tells us something? And what it came down to was that organisms like Halothiobacillus, Thiomonas, Thiovirga, these guys that can do complete socks, right? They're the ones, if they're present, then you're going to see more generation of acidity and sulfate. When we see the green guys, and particularly Thiobacillus, he's our star slow guy, tends to be present in most of the mines. They're doing this RDSR. That's when we see a delay in acidity generation, because essentially what they're doing is recycling sulfur in these lower oxidation states. And so that really, started to give us hope, because we saw a coherent picture starting to come together of which organisms are present. They're present across all the mines. They're similar. What are they doing? We now saw that we could see that. What we wanted to understand was what controlled whether we had fast or slow, because obviously that's when you start to have levers that you can apply to essentially manipulate the sulfur oxidizing bacteria to get what you want. And so we have some field insights, and we're still working on this. This is a paper that came out just earlier this year in Nature that started to look at the influence of particularly of oxygen and thiosulfate in how we were seeing zonation of this fast and slow organisms in one tailings pond. And so what you can see here is that we've got increasing depth as you go downstream. As you look across, you see that acidity generation is highest in the surface waters. You also see that thiosulfate concentrations increase with depth. So deeper you go, you have more thiosulfate, but also that oxygen decreases. And when we look at our fast and slow guys, we see Halothiobacillus, is our star fast guy, limited much more so in their activity to the surface waters where there's more oxygen, whereas Thiobacillus, the slow guy, starts to take over as oxygen decreases. And what this really allowed us to do was link sort of characterization and zonation in terms of environmental geochemistry with which sulfur oxidizing bacteria are present and which pathways are operating. And so that really was important, because now we're starting to think about how do we monitor. We now can think about not just geochemical indicators, we can think about biological ones that might actually be useful indicators that we could use. And so when we started to put this together and organize across all of our mines, we had intuitively seen, looking at the data, that pH seemed to be an important control on where we were finding not only SOB in general, so polishing ponds that tend to have very high pHs, we don't tend to find organisms present. High pH is typically difficult for most microbes. But if we looked at sort of lower pHs, that's where we tended to see the fast guys. And more circumneutral pHs tends to be where we see the slow guys. And this tends to be most tailings ponds. So the likelihood that these slow guys are present is quite high, and that they are cycling sulfur and generating some of these other compounds. So I think that the future is very bright for the development of biological indicators, and we're working on that right now. And again, to make them as simple and cost effective as we can. And then we have to think about treatment, right? And treatment doesn't always have to be fancy. I think it can just be simple interventions. So, when I think about essentially, if we're thinking about a mine context, if we think about fast guys, I think they're desirable to promote that on site, which ironically is the opposite of abiotic tailings management, right? Pump up the oxygen, try to promote those fast guys so that they process any of that reactive sulfur on site, deal with the acidity and sulfate if you need to before you discharge. They're undesirable off-site because they will lead to acidity and all of those impacts we don't want. Generally, the slows, you don't really want them in either case. Because they're going to essentially alter sulfur. They're going to generate more of these compounds that could either cause increased effects or delays in effects and persistence of the ability of the fast guys to hop on when conditions are favorable. And so one of the things we've been thinking about is that sort of inserting another system between sort of secondary, or if you only have one tailings pond, and the polishing pond, is think about a way to promote fast SOB, right? So you take in that water, you increase oxygen, get the pH range down, maybe to 6, and really allow for, and you could seed it, right, as well with the fast guys, but really try to get them to process most of that sulfur out before it goes through treatment. And this would be simple things, but then there are other ways we can think about which are more fancy in terms of treatment, where we start thinking about biological treatment, where we're manipulating either at tailings pond scale or in sort of controlled scaled reactors to really pump water through and sort of get them sort of going through the bugs that we want. And so, I think it's been a bit of a whirlwind, and I'm happy to take questions now or after. But I think, as we start looking towards moving from mine water management to mine water stewardship, really the industry needs to take advantage of these tools. Mining waste contexts are biologically reactive systems, and they're not well described. The good news is that we have the tools to actually investigate, and I think we have the strategic insights now to figure out how to do that quickly. And so geochemically, I think there are always ways we can improve, sulfur in particular, but I think we've shown that, bringing in some fresh ideas and different methods really could be efficient for the industry, but also that we might be able to use some of these microbial new knowledge discoveries we have now as ways to essentially predict impacts and therefore establish the risk. But we also can think about them to prevent impact and eliminate the risk. And I think that this idea of new tools is not just an academic one. I think as we know, these tools are being deployed and used in so many other sectors to great advantage. And I think the industry just needs to recognize this is just another suite of opportunities to really increase their efficiency and increase their stewardship. And so just to sort of give you the top 5 takeaways, right? We have definitely a mining wastewater, at least for sulfide rich ores that are being milled, definitely a sulfur oxidizing bacterial microbiome that's distinct from other sulfur rich contexts. We've identified that fast and slow, so complete SOx versus RDSR pathway guys, and the implications for sulfur risks. We started to constrain. I don't think pH and O2 are the only ones that are going to control this or continuing to work on this. And we know that, in fact, microbial signals, and we've been doing some of this beta testing with Hud Bay, on-site signals with fast and slow organisms, so percent abundance, is correlating to observed toxicity test outcomes in the receiver. So I think the power of this is really demonstrated. This is, one of the first cross-mine studies that's done this kind of genomic integration investigation. And I think it's massively underutilized. And I think there's huge opportunities to move from reactive water management with limited tools that are really not gearing mines to be 100% efficient, and really start moving to being proactive and adaptive and decreasing their risk. So with that, I'd like to thank you. And I also like to say, obviously this work is carried on the shoulders of many, many people. And it's not just myself who's been involved. And I'd just like to say thank you. And if there's time and people have questions, I'd be happy to answer them. Thank you.
So it looks like Bernard has a question, and I'll see if I can open his mic because I think it might have a great conversation.
Sure.
There we go.
Well, Marcia's, oh, perfect.
Okay, so you guys don't have to read my question. I can read it myself. Well, first of all, super interesting. I didn't know this was going to be about thiosalts, and I'm right into thiosalts, and I didn't know these studies were going on, which is great. My first question is, when you were saying that you're measuring the thiosalts by difference from sulfate to total sulfur. I've been analyzing sulfate and sulfur for 30 years. And
fantastic. What a data set.
Yeah. And I have so often seen that the sulfate was being done by ion chromatography,
Right.
total sulfur by ICP-MS,
Yep.
and the sulfate was too high to account for the total sulfur. So there was more sulfur in sulfate than total sulfur would make it possible to be. So if you're doing your thing if you're getting some stuff like this and you're doing your thiosalts estimates by difference, that would really complicate things.
Yeah, and so we worked a lot. You probably know Simon Apte, who is sort of the lead CSIRO analytical chemist who's been working with us on this. And so we do total sulfur using ICP-OES. And I think that we have had a couple of instances where there has been, I wouldn't say hugely different, but basically your numbers are indiscriminable between total sulfur and sulfate. And to your point, we would argue that that just means that all of your sulfur is essentially sulfate. I have not seen, and we have collected, because we also do work in the oil sands, and so we are sampling there. So we probably have a data set of about at least 1,000 data points by now. And I haven't seen anywhere our sulfate numbers are hugely over our total sulfur. So I'm wondering if it's resolution of the infrastructure. And do you think would you say in earlier years there was more discrepancy, or do you think it's a consistent?
For me, I've seen differences often above 10 or 15%
Okay.
too much. And these are cases where often there is no thiosalts.
Okay.
So it really is. So for me, what I've been doing for the past years is using ICP only for total sulfur, because otherwise I don't trust IC.
Yeah. Okay. Yeah, and and I'm sure you do this as well. And the sulfur piece, I agree, and Simon and I talk about this quite a lot. So we typically do unfiltered and filtered samples as well, and we have been playing with dilutions, right? Because if there's a matrix effect, because a lot of our samples, especially, well, it's in fact all of these systems where there's sulfur, there will be elemental sulfur present. And so if you're doing unfiltered versus filtered, we often see differences there as well. But yeah, I think that it's something it's been on my mind for a little while, and maybe it's something you guys could think about organizing, because I think that the sulfur speciation and analyses, and just how do we start drilling down to what's an effective and accurate methodology to really track this would be really important. But we haven't seen that large a difference, I would say, in the data we have. Mind you, it's not 30 years worth of data.
No, it's not. And I don't keep all that data.
Yeah.
Well, I do have it. I don't do the analysis myself. I'm a water treatment guy.
Right.
But I have another question though.
Sure.
Because I've also been involved in thiosalts for about 30 years. Started with Brunswick Mine when I was at Noranda Technology Center, and I've been involved with Raglan in Portugal as well, where they had thiosalt issues at Cemincor. So I really loved your presentation. But you mentioned, okay, the slow oxidizers and the fast oxidizers.
Right.
Now, what I've seen is a huge temperature dependence. At Brunswick Mine, for example, you wouldn't have any thiosalt issues during the winter at all, because the thiosalts would make it right to the ocean without oxidizing. During the summer, you wouldn't have any issues with thiosalts because they'd all be oxidized before getting to the lime treatment plant, and then it wouldn't be a problem. In the spring and fall, it wouldn't oxidize. By the time it went through this treatment plant, would oxidize in the environment, acidify the receiver. So in this case, it's not really so much about the genomics, about the kind of bacteria. It seemed to be straight about the temperature.
The temperature, and I think to your point, and particularly spring freshet, depending on how much water you get and when you get it, that plays a role. I'm not sure that I agree But I would also say that I'm not sure I have enough data to say, you know, with 100% confidence, I believe that, you know, bacteria are important. I know they're there. But to your point, you know, if you're seeing that essentially you've got times where thiosalts are making it through and they're discharged to the ocean, so it's not an issue for the mine. It speaks to the fact that they've either hung around, either that sulfur oxidizers were not active, or in fact they were, and they were the slow guys, right? And they were essentially recycling sulfur in these lower oxidation states. The fast guys were obviously, in my view, and some of it can be abiotic, but what we're seeing is that that biological signature is really important. And so, and we're trying to figure out right now, what is the lever? Because across mines, the slow guys are present in all the samples we have. The fast guys pop up from time to time. And so what we'd like to know is what precipitates that shift. Like we know oxygen seems to be one thing. One of the mines where we saw the fast guys come up had actually had an operational shutdown. So they had not been discharging tailings. And so that really sort of spoke to that. We thought it was a salt thing, but also potentially an oxygen thing. But I think you're right. I mean, there's way more that we need to tease out. And I think that it's just, we've been, as is typically the case, we're called in when mines are having problems with their current models, their current monitoring, their current treatment. So, you know, we're called in because it's pretty obvious the tools they have aren't working. And so, but what I think would be far more powerful is to start establishing baselines with mines where things aren't going wrong, right? Who's there? What's happening there? And to your point, I think especially with climate change, we need to absolutely layer into this water delivery, quantity and when, right? Because the timing of that is changing all over the place. And so you know, there's a lot more parts to this puzzle. But you know, in my mind, sort of 30 years on now of trying to sort of build this story of understanding the biogeochemistry of the system, genomics is absolutely going to inform you quickly and efficiently. Is this something you need to consider or not, right?
Oh, absolutely. I think what you're doing is great work and should continue for sure. I'm just, so in your test work, have you tried to see a temperature influence or just always working at?
No, so we are, in fact, it's definitely something, because one of the things we also see is in the winter, so we do under ice sampling when we can get there. And in fact, we've got better access. We work with Syncrude, so we've got better access in the winter on the oil sands, but we've seen it when we've been able to get access and get samples under ice in base metal as well. It's a different community. So it's a different community of bugs. And so there's definitely a temperature, there's an ecological temperature, I think, lever in terms of which organisms are present. But what we don't yet have enough understanding for me to confidently say is, you know, are they always slow? Are they fast? We're not quite sure yet. But it's definitely something we're interested in understanding, because what's happening under ice, processing, right, then kind of sets you up, I think, depending on what your spring freshet is like, for what the summer may start with, at least.
Cool. So final comment.
Yeah, thank you, Bernard. We have another question.
Yeah, it's not another question. You got to present this at ICAR 2024.
Oh, I'd be happy to do that. I'd be happy to do that, and hopefully we'd have more data. You know, I think there's so much we can benefit too from the insight from everyone who's working in the industry. You know, the first thing I would say is, I'm not a microbiologist by training, so you know, I'm a geochemist. But there's so much I think we could bring into this and inform in a discussion. I'd be delighted to do that. So and hopefully we'll have a bit more to sort of fill in some of these other blanks. But I think it's exciting. You know, in my view, I think there's 3 things that really excite me in terms of practical tools. One is the diversity of organisms we find is pretty small compared to what you might find in a soil, right? So we're not dealing with thousands of organisms. We're dealing with kind of a smaller diversity, and that makes sense because these systems are not, you know, they're more extreme compared to a soil or a lake. Secondly, that diversity is not only is it tractable, we see the same kinds of organisms across, you know, these 4 mines and in their receivers. So there are links there. And then we see this, very clear pattern. If you've got sort of the complete SOX, the fast pathway operating versus the slower RDSR pathway operating, we know what the outcomes are, but we also tend to see that they almost alternate in dominance in the community. So it speaks to what causes the flip. And if we could figure that lever out, that's when I think we start saying, this is what we need to do to implement sort of just a strategy. And it could be, simple as increasing oxygen or decreasing pH that will promote the fast guys, or, whatever way we want to go. So when we talk about biotreatment, we're also thinking about very smart interventions that could be very simple. But the monitoring would also be simple. We could probably do it with 16S. And so that's what we want to do too, is figure out, who are the guys that you need to know, and then can we do it in a way that looks at ratios of abundances, because a 16S is a few $100s and can get it done very quickly. So we could, that's something from a monitoring perspective, I think as we start to build that out, could be very useful. But, I always gain having discussions with people about this, because there's always broader insights that come in. So yeah, I'd be happy to come and talk. Where is ICARD 2024?
We'll be advertising it in fall of 2024, Bernard.
Oh, it's been advertised for a bit. That the abstract submission deadline is November.
November. Okay, I'll check that out.
It's going to be in Nova Scotia this year.
Okay.
In September 2024.
Okay, perfect.
Great questions, great feedback. I just want to give Benoit a chance to ask his question. I can. Oh. I'm having trouble with his mic. He asked, genomic question, do you see any organisms with a partial SOX pathway? How do you think this would affect the classification between fast and slow S oxidizers?
It's a really good question. So most organisms, and even like Thalabacillus, let's see if I can just go back to the slide real quick, have the incomplete SOX pathway. And the way that you actually determine are they complete SOX is to look for this SOX CD. If you don't have SOX CD, they can't actually take it all the way to sulfate. But you'll often see, and you can kind of see by the boxes here. So these all represent the different genera that have those genes. And so what we see is that, you can see the fast guys, 100% of them have it. Whereas when we start looking at the RDSR pathway, really, it's really limited to who has that. But the incomplete SOX versus complete SOX, what you're looking for is SOX CD. I hope that answers the question.
I'm sorry, I can't unmute him for some reason, so I apologize, Benoit. And I hope I pronounced your name right.
I think we actually skipped over to Catherine's question. I have one from Benoit about iron oxidizing bacteria. So I'll go ahead and read if there's some troubles with the microphone. Awesome stuff, guys. Are you conducting the same research for iron oxidizing bacteria, and biofouling? There's a lot of potential applications there as well.
Sure. So we're not looking at iron. We collect data on iron, but iron concentrations in some of our systems are pretty low. And there's been lots of work on iron oxidizers, as you probably know, but it hasn't been a key focus, particularly in the thiosalts work. But we have been looking at them in our oil sands work, because that's a pilot pit lake, and we're trying to understand the complete biogeochemical cycling that would impact oxygen. And so we're looking at sulfur, carbon, nitrogen, and iron. So in these systems where we have higher pHs typically, iron oxidation is pretty quick. So windows where iron oxidizers could actually get in on the action tend to be limited or just not there. So unlike ARD, where you slow down those oxidation rates and allow, once you get down to acidic pHs, and allow for bugs to take advantage of it, it's not quite the same. But you're absolutely right. These genomic technologies, and it's funny, I think a lot of technical people are very comfortable with pH probes and things like that. It's the same thing, right? These genomics technologies are definitely sort of applicable. I think it's just understanding what they can tell you and how to integrate it into sort of a geochemical characterization. Went off the.
Thanks so much, Lesley. We're just coming up on 2 o'clock right now. I actually have a personal question that I'd like to ask, but since
Sure.
we are right at 60 minutes, I will point to the handout that Marcia kindly shared. That'll have Lesley's contact information, a little more information about the ESRS upcoming events, and some links with some more information about ESRS if you're interested in reading more. So as to my personal question, I was wondering if you see more applications of genomics to waters for like internal or closed loop processes, or waters destined to be released for environment. And I think one internal that I'd be interested in is like thiosulfate gold leaching. I know there's a lot of sulfur consumption there. Could genomics help for that?
So what's surprising, and we have grabbed a couple of samples from the mill at a couple of our sites, and there are bacteria in there. So, what they're doing and how active they might be, not really sure, but absolutely, right? So, cyanide is obviously one that comes to mind when we start thinking about gold. And we know that we have bugs that can do both ways, right? And are happy in that. I think, we've always come in on the environmental side, simply because that I think is a much more, it's a growing risk, and it's increasingly, influencing reputation of mines. And I think environmental managers are increasingly saying our tool belt just isn't working. We need better tools if we're going to adapt and sort of manage these systems more effectively. But I think in terms of the sort of thinking about processing and milling, absolutely, I think it's worth having a look. That, to actually investigate, are there bugs there? If so, what are they doing? Is absolutely of use, because that's not a really expensive thing to look at. And you're always looking at, are there ways, and also, what I'm always thinking about is if you have organisms that are living under those conditions, we might be able to take them and deploy them somewhere else, right? They become almost a treatment that you could use, or you find your superstar, right, that's really able to leach. It's like thinking about chalcopyrite. And so I think the applications and the opportunities are really huge in terms of what could be looked at.
I really appreciate it. It is interesting to see all these new technological developments and how genomics can actually be applied into them as well. That looks like all the audience questions that we have. Let's see, Marcia, are there any questions that you had in mind that you'd like to add, or?
I thought it was a great presentation. I don't want to take much more of Lesley's time or everybody else's time, but I would really like to extend gratitude that you were willing to present today. It was very informative, and I look forward to another webinar and more information sessions from you in the future, Lesley.
Thank you. It was my pleasure. Absolutely. And great questions, great discussion. And anybody who's interested, just feel free to follow up. I'm happy to chat.
Thank you so much, Lesley. And thank you everyone for attending today.
Thanks, everyone. Bye.